much for the invitation. So since I'm the first speaker here, I will first then briefly present the sodium potassium ATPase, the ion pump, as an ion pump. It's a transport, as you know, then the uses the ATP hydrolysis, the energy from ATP hydrolysis, to transport sodium out of the cell and potassium into the cell, creating then an gradient that allows for ion-coupled uptake of cell nutrients such as the glucose and amino acids at no extra cost. Very energy efficient. The pump was discovered by Jens Christian Skov in the 1950s. About then 40 years later, evidence started to emerge from the Toledo University in the US and from our lab at the Karolinska Institute in Stockholm, that sodium potassium ATPase is a multifunctional protein, and that it may also act as a signal transducer. Our studies have been carried out in epithelial cells because we've been interested in nephrology, and as you know, about 80% of our cells are indeed epithelial cells. So the sodium potassium ATPase then has, in contrast to most other ion transporters, then a binding site for a substance that is present both in plant and in animal cells. It's the cardiotonic steroid, the group of the substances, cardiotonic steroids, and among the cardiotonic steroids, one of the cardiotonic steroids, ouabain, is then identified in the blood of mammals, including then humans. Here you see then my second slide, which shows then that the sodium potassium ATPase is not only a pump, but also then a signal transducer. And then, as I mentioned, then our group has been studying the sodium potassium ATPase as a signal transducer in epithelial cells, since we are also from the beginning very interested in kidney function. Okay, so let me see now. So about one day, in the late 1990s, Per Ullén, who was then a PhD student in our lab, he is now a professor at the Karolinska Institute. He came rushing into my room and said, "You have to come and have a look. I added ouabain to these epithelial cells, and when I was recording calcium, and what I saw was calcium oscillations. Could that be possible?" And I came in, and yes, indeed, did he then record calcium oscillations, and at the same time in then cells that were given ouabain from very, very low concentration, 10 nanomolar, up to 500 nanomolar, and regardless of the concentration, we saw an oscillatory calcium response that was very similar regardless of the concentration of ouabain. Here, so the frequencies was the same in the then, regardless whether we gave 50 micromolar or 500 micromolar of ouabain. With higher concentrations, then we inhibited the sodium potassium ATPase and just got an extended increase in the calcium recording. Okay, so this was very exciting because the calcium oscillations is a highly specific signal, and it attracted then the interest from Professor Miko Shiba at Tokyo University, who is the giant within the calcium research field and the discoverer of the IP3 receptor. And he told us that calcium oscillations then needs then calcium release from the IP3 receptor. So the sodium potassium ATPase must then activate the IP3 receptor. And together then we did a study here to identify the site of the sodium potassium ATPase that might interact with the IP3 receptor. And our candidate was the N-terminus and the N-terminus tail, because the N-terminus is, in contrast to the rest of this protein, not involved in the transport of sodium and potassium. So maybe it has some specific role. So together with then a postdoc from the Miko Shiba lab, then we were able to identify a site on the N-terminus of the sodium potassium ATPase that directly interacted with the IP3 receptor. So calcium, then we conclude IP3 receptor must be involved, but the problem is not involved alone to then create the calcium oscillations. Calcium, when you activate the IP3 receptor, calcium leaves the endoplasmic reticulum, calcium goes up, and then it leaves the cells. But the IP3 receptor is in some way that is not yet quite clear. Then it will be activating an influx calcium calcium channel called STIM, and calcium then re-enters the cell and back to the IP3 receptor, and thereby then you can create this oscillatory then signal. Calcium out into the cell, leaves the cell, back into the cell via the STIM, and then back to the endoplasmic reticulum, and it's repeated. Calcium oscillation makes the otherwise a single calcium peak is a very unspecific signal, but calcium oscillations activate several calcium binding sites with different on and off time, and that makes this signal highly specific. So it will only activate a few proteins, and among those proteins we were then looking for proteins that had been reported to be activated by an oscillatory calcium signal, and we found calmodulin and we found NF-kappa B. Now, interesting, then these two proteins are both involved in the regulation of apoptosis. So that raised the question, could ouabain regulate apoptosis? Well, so we decided to test this, and then we used a cell line, an epithelial cell line, where we then also had in one types of cells we had then deleted the N-terminal tail of the sodium potassium ATPase. So it was wild type sodium potassium ATPase that was expressed in the cell line, or it was a sodium potassium ATPase that lacked the N-terminal tail. So black and wild type, and gray then lacks the N-terminal tail. And what we did with these cells, well, we exposed them to serum starvation, to a classical way to induce apoptosis. And then with serum starvation, we got a striking apoptosis. Then we repeat it in the presence of ouabain 1 nanomolar or 10 nanomolar. And in both cases, then we then completely inhibited the apoptosis in the cells expressing wild-type sodium-potassium ATPase, but had no effect in the cells that then lacked the N-terminus of the IP3 receptor, and therefore could not forward the signal from the sodium-potassium ATPase to the IP3. So this convinced us that then the calcium oscillations, these that we could observe that were as then induced by ouabain, then had an effect on apoptosis, that they protect the apoptosis. And that's then extremely interesting because, as you all know, then apoptosis is our best defense against cancer, but it's a process that has been very rigidly controlled, because in many cases of, say, for instance, the neurodegenerative diseases, or in the complications from diabetes, there is an inappropriate apoptosis. They start off to get into apoptosis, you load the cells, and then you'll have then a further progress in these diseases. So a very important clinical question is, could ouabain protect from inappropriate apoptosis? That would be maybe a very, very helpful in the drug in the future. So we set out to study this. But first, we wanted to confirm then whether this then ouabain acted as an inhibitor of apoptosis in different kidney diseases that led to progressive kidney failure. And we spent many years doing this, because it was not that easy to convince the ATPase people that ouabain also had a signaling effect. But during these 10 years, we could then report that in the an embryonic, if the embryonic kidney then is exposed to, if the embryo is exposed to malnutrition, it will have adverse effects on the kidney. If, however, then we treat the pregnant rats with ouabain, then we completely protect from these adverse effects. So that was very promising. Then we continued with the different types of experimental kidney diseases, studied then in mice. Hemolytic uremic syndrome causes then renal acute renal insufficiency, and it's caused by apoptosis of the renal cells by the factors that then produce the hemolytic uremic syndrome. And in the study then published in the American Society of Nephrology, we could demonstrate both then in cultured cells and in mice that were exposed to the shigatoxin to produce hemolytic syndrome, that ouabain seemed to then to completely protect from apoptosis and also protect from renal damage in hemolytic uremic syndrome. Then next, we also studied other types of proteinuric kidney disease mice, the same results. We found protection from loss of kidneys, from the kidney destructions, and protection of renal function. And both now these are all relatively uncommon conditions. If this should be of big general importance, maybe we'd also like to look at some more common disease. And the most common cause of renal insufficiency are these diabetic complications due to exposure to high glucose concentration. Um, so we then specifically looked at the effect of high glucose on kidney, on proximal kidney cells in primary culture, and cells that were cultured for a couple of days were treated with glucose. Normally the kidney cells are exposed in culture conditions to 5 millimole glucose, and then we increased it to 10 or 20 millimole. And for 2, 4, and 8 hours, we saw a progressive increase in the apoptotic index. And at 8 hours, there was a huge then increase in the apoptotic index, but that was completely abolished if the exposure for high glucose was combined with exposure to ouabain in low concentrations. So this then suggests that in the future, then ouabain may be a very useful drug to protect from apoptosis. Before then getting it to developing it into a drug, we have to learn much more about its molecular effects. And so we started to look at the molecular mechanism of apoptosis, which then this slide shows you. So let me then briefly remind you that there are 3 types of proteins that determine the apoptotic process. 2 that then can produce apoptosis, and 1 defender. The pro-apoptotic protein, the most common, is BAD, the apoptotic protein BAX, and the anti-apoptotic Bcl-xL. And under then control in our cells, so then these proteins have to be strictly controlled to avoid inappropriate apoptosis. So the pro-apoptotic then proteins, the pro-apoptotic protein BAD and other pro-apoptotic proteins are kept outside the mitochondria in tight connection with then the signal transducer, the multifunctional signal transducer act. The anti-apoptotic protein Bcl-xL then protects the mitochondria, is the protector, protects the mitochondria from apoptosis. And the apoptotic protein BAX is outside the mitochondria, could enter, but in this then the BAX, in the presence of then Bcl-xL, BAX will leave the mitochondria, so it will never accumulate on the mitochondria with a healthy Bcl-xL. So what happens then when you start the apoptotic process for some reason? Well, then the first thing that happens is that the pro-apoptotic protein BAD will leave ACT and enter the mitochondria, where it binds to Bcl-xL and then inhibits its effect to prevent BAX from entering the mitochondria. So Bcl-xL is now inhibited. BAX enters the mitochondria then and makes hole into the mitochondria membrane. Cyclists and other factors are released from the mitochondria and they leave the mitochondria, activate caspase, which will, and other substances that will that are toxic to the cell by caspase, by inhibiting a number of essential proteins. So if we want to therapeutically then inhibit apoptosis, we need to have a drug that acts before BAX enters the cell, because then it's too late. So the question, can ouabain be used as an anti-apoptotic drug? Then only if it acts before BAX penetrates the mitochondrial membrane. Now, how do we try to answer this question? Well, here we are lucky because we have the possibility to use the super resolution STED microscopy, which gives a resolution of 50 nanometer. And what you see here are then mitochondria that are visualized with a fluorescent dye, and then the cell is stained for, in this case, for BAX, the apoptotic protein, and Bcl-xL, the anti-apoptotic protein that under controlled conditions protect the mitochondria. And so then the bioengineers in our lab then uh, uh, helped us to develop a method to calculate the number of, or to calculate or to estimate the amount of BAX that would enter the mitochondria. So what they did was to see how far from the from the Bcl-xL is the BAX in here. We'll go back here. So how far from Bcl-xL in the mitochondria could how many BAX molecules would we see that are attached or close to or very close to Bcl-xL on the mitochondria? How many are within 50 nanometers and within 25 nanometers reach, within 50 nanometers reach, and so on up to 200 nanometers reach? Up to 200, then there is still a risk that they are in the mitochondria, that BAX would be in the mitochondria. So this out then to the right here, you see the results here from a study, from the study when the cells were exposed to high glucose for 4 or 6 hours. And up the upper panel are the controls about that shows the number of BAX that are within 50, 25, 50, 100, 150, 200, 250 nanometer away from Bcl-xL. And when you and the next panel is after 4 hours after high glucose, and as you can see, there is a huge increase in the number of BAX that now are very close to the Bcl-xL. And that is, they are in the mitochondria, they are close to the Bcl-xL, and Bcl-xL will be then inhibited and can no longer protect the mitochondria from penetration by BAX. And at 6 hours, even more BAX have entered the mitochondria. And that's where you then can note that there is a high level of apoptosis going on. So what happens then if we add ouabain to the cell? So with BAD, yes, to the left, then we add the BAD in the presence of high glucose, and to the right, we added in with high glucose in the presence of ouabain, and you see that there is definitely much less BAD that is close to the Bcl-xL. BAD has not then moved into the mitochondria and associated with Bcl-xL to the same extent in the ouabain treated cells. Seems then that ouabain then protects BAD from entering the mitochondria. And the effect of BAX is even more striking than high glucose. Remember, then a lot of BAX had entered the mitochondria after 6 hours after high glucose treatment. But in the presence of ouabain, then we almost completely prevented the transfer of BAX from the cytoplasm to the mitochondria. So then to summarize then the findings is that ouabain will inhibit bad transfer to the mitochondria and thereby inhibit the association between BAD and Bcl-xL that makes Bcl-xL, it makes it possible for Bcl-xL to protect the mitochondria. And then gives the way for BAX, gives the possibility for BAX to freely enter the mitochondria and then results, which then result with cell death. But what we have shown now is that ouabain protects then the pre-apoptotic bed from entering the mitochondria, and therefore then ouabain, we conclude that ouabain would inhibit apoptosis from its point of no return, which would be an essential criteria for a safe anti-apoptotic drug. So at this point then, many years of work, we come to the conclusion that ouabain activates calcium oscillations and that ouabain protects from apoptosis by bad inhibition. But what are the molecular mechanisms? Well, one answer to this would be then to perform a phosphoproteomic study to then look for all the proteins that could be regulated by ouabain via phosphorylation, dephosphorylation. And we had then a very talented PhD student, Elena Panizza, who worked with this project for a couple of years together, and then also with her mentor, then Janet Leshti. And what this then complicated then shows is that to the left is that demonstrates these little black dots are the proteins that were identified in this study, 7,000 proteins that had phosphorylation, dephosphorylation sites. But untreated, we were not looking for any change in phosphorylation. So then the untreated, the cells were studied at first twice with 10 minutes in between them, and there was no change. But if we exposed the cells to ouabain and then waited for 10 minutes, then we saw a huge change in the stage of phosphorylation and dephosphorylation. To the left are the red to the left are dephosphorylated proteins, and the red to the right are phosphorylated proteins. And that was after 10 minutes of ouabain treatment, then this change from then appeared. So ouabain we were surprised to see the enormous effects that ouabain had on so many proteins in the cells. I should mention here that we used a COS cell line that is derived from monkey kidney cells. Okay, then we next then we also studied them of 20 minutes after we had applied ouabain. And as you can see here, there is a difference between the 10 minutes and 20 minutes, which means that a lot of things are going on in the state of phosphorylation after you apply ouabain also continuously. So from untreated to treated, we see huge changes in phosphorylation, dephosphorylation states of the proteins. And from 10 to 20 minutes, there are additional huge changes. So Which are the proteins here? We've identified a number of very interesting proteins that will be then studied in the future in this publication of the FEBS journal. And they are all, you have a list of them in the supplement. But important for this study, first thing is that we found that ouabain regulates both IP3 receptor and the STIM receptor. The two receptors that you may remember then are responsible for the oscillatory calcium response. So ouabain then creates this difference in the permeability probably of the IP3 receptor and the STIM receptor by then changing their state of phosphorylation, dephosphorylation. We were, of course, interested in regulatory proteins. And then the kinase, we found then that looking both for protein kinases and protein phosphatases. So among these different groups, we found that kinases were specifically enriched among the ouabain regulated phosphoproteins. And among this group of kinases, we identified the kinase 1, KK1, and the KK2B that have both been implicated in the regulation of ACT, that they were both ouabain regulated. So then, this raised the question here. So we have this again going back to the scheme for apoptosis. Then what we didn't know is what is the relationship, what regulates the relationship between ACT and BAD, what releases BAD, so that BAD then can be taken up by the mitochondria. We'll move to the mitochondria. Could this then be the activating can ouabain? That we asked the question whether then mitochondria we could activate by activating CAM kinase 1 and CAM kinase 2B. And so we did what we did then was to decide to downregulate these two then CAM kinases, and one is CAM kinase 1, and so we again had cells exposed to high glucose in the absence and presence of ouabain, and as expected, ouabain inhibited from the apoptotic effect of high glucose. But when we had downregulated CAM kinase 1, then completely downregulated CAM kinase, and then high ouabain had no longer any effect. So ouabain must act when CAM kinase 1 must be one of the then CAM kinases that ouabain is regulating. The same thing with CAM kinase 2, gamma, then high glucose, then high glucose caused apoptosis, and ouabain protected from apoptosis, while when following downregulation of this CAM kinase, then again we completely then completely lost the protective effect of ouabain. So with this, then we have then identified the ouabain bound exerts its protective effect by activating these two CAM kinases. And these two CAM kinases play a very central role in that that makes bad stick to act and prevent the activation of a death signal. So to summarize then, sodium potassium ATPase is a multifunctional protein. Ouabain, an endogenous sodium potassium ATPase ligand, triggers an oscillating calcium signal in epithelial cells. The calcium oscillation signal regulates two calmodulin kinases identified in a proteomic study. The calmodulin kinases protect the binding between ACT and the pro-apoptotic protein BAD. Deactivation of these proteins initiate the apoptotic process. And as ouabain will inhibit the apoptotic process before the point of no return, it fulfills the criteria for an anti-apoptotic drug and should be useful in diabetic nephropathy and other progressive kidney diseases. And finally, the phosphoproteomic study has revealed that ouabain regulates numerous proteins of vital importance for regulation of tight junction and between epithelial cells and cellular proliferation. I'll just show you a little bit of this here, because among these 15,000 phosphosites that were then detected on 7,000 proteins, this then, as we see it, will create a gold mine for future studies of sodium potassium ATPase signaling. And as I just briefly mentioned, then we found that, which we have not yet studied in detail, that ouabain regulates protein as involved in regulation of cell adhesion. Numerous proteins that are involved in cell adhesion are regulated by ouabain, and numerous proteins involved in cell proliferation are regulated by ouabain. I think that this opens up for many interesting studies of the role of ouabain for cell adhesion and cell proliferation. Just to suggest some suggestions here that that the ouabain, that the epithelial cells that we have studied then are held together by tight junctions, and one of the questions is that, and in cancer, then you cell dissociate, and because you lose the effect of the tight junctions, and would ouabain maybe protect then from cancer metastasis, because it's the cell dissociation that results in cancer metastasis. So could ouabain have a cancer protective effect? One major question to be addressed in the future. And with this, I will end here and just then mention the people that have been involved in this study throughout the years. Then they have all then made very important contributions. All these PhD students and postdocs listed here then have the great advantage to work with a physicists then were interested in medicine, Hjalmar Brisman, throughout the years. And on the top are the three people that have very much inspired to this study: Jens Christian Sko, from whom I learned a lot about the sodium potassium ATPase; Paul Greengard, then the discoverer of the role of dopamine and how the dopamine via phosphorylation regulates then brain function. From Paul, from a long time then collaboration with Paul, I learned the importance of phosphorylation, dephosphorylation regulation. And then Professor Miko Shiba in Tokyo for then along as and in collaboration on the role of calcium oscillations.